So the first thing I'm going to do is do some of the housekeeping up front, because as you notice, the other name on that first slide was Allison Van Eenenam. So when we get towards the end, I'm actually going to turn it over to Allison and have her show you a little demonstration of a program that's part of this grant that I think will be really interesting for you. So the project team is obviously a fairly large group of people. The grant is headquartered at a University of Missouri, but includes a wide variety of other people and other institutions. So I also want to cover a few acknowledgements up front. I usually do that at the end. But if you enjoy hearing about this and are glad your breed association is involved and they were one of the ones that assisted in sequencing efforts, please make sure and say thank you to them. Also got a lot of reproductive information on a bunch of heifers from the Missouri Show Me Select Replacement Heifer Program, as well as the Missouri Angus Association and Circle A. And some funding from a couple of different USDA grants went into this. And as a couple people mentioned, I will tell you just a little bit about the GDP F250 chip that was designed partially to augment this project. Notice you're going to talk about it quite a bit tomorrow, so I'm not going to belabor these points a lot because you're going to hear about it again, but I will give you a brief overview of that product. So first, let's talk about the project goals. So we've seen a lot of tremendous gains in terms of other production traits, right? Growth, I think milk production was talked about earlier today. So we've seen a lot of pretty dramatic changes in genetic trends in those types of traits. But I would argue we maybe haven't seen quite as large a gain as maybe we would like in fertility-related traits. And it's really no secret that reproduction is an incredibly economically important trait within the cow herd, and advancements in that trait would increase profitability. But at the same time, we want to do that in a way that doesn't sacrifice performance and other economically important traits and improves overall profitability of the cow herd. So really, we're looking at maximizing the number of females that conceive early in the breeding season and maintaining those pregnancies that are achieved. So the strategy that this project took was really to try and identify lethal alleles that may be causing early embryonic mortality. Okay, so a lethal allele would be a broken gene that is essential for life. Okay, so if you don't have at least one functional copy of one of these alleles, the result is pretty bad, right? You're dead. So I like to make this analogy with coffee, right? How many coffee drinkers do we have in the audience? Quite a few, right? So if you don't get your morning coffee, are you very happy? Right, not very happy. Okay, so basically this analogy works because we have two chances to get a functional gene, right? Just like we would prefer to have 2 cups of coffee in the morning, right? We're going to be much happier if we have 2 cups of coffee as opposed to 1. So the absolute worst scenario is if we don't get any coffee at all, just like if we don't get any functional genes from either mom or dad, right? So we've all seen the effects of these lethal alleles and other types of genetic abnormalities, right? That's not going to be anything new. AM, NH, all of those. But we also have early embryonic lethals, and the challenging part with these is that we don't actually see the effects of them as obviously, right? They're going to show up as open cows or cows that are slower to breed back, or maybe cows that abort and come back into heat at some point in that breeding season, okay? So these are going to be harder to see unless we have very detailed sorts of fertility records. So let's talk a little bit about what lethal alleles might be, because they can be a lot of different things. And in talking about the F250 and talking about causal variants and functional variants, all of these things play a role. So I'm going to try and define them just with a brief overview here. So let's say we have this fictitious gene here, and it's got 5 exons. Okay, so the exons are the part that actually go into making the protein. That's part of the protein. Okay. So all of that is read in sequences of 3, so 3 bases at a time, and that tells us what that sequence of amino acids is going to be in that protein, okay? So if we generate one of these different final proteins, right, we think about these things as 2-dimensional molecules, and that's basically what I laid out here, but all of these things operate in 3-dimensional space, right? So if we change the structure of the protein by changing the amino acid sequence, a lot of times we can have some sort of disruption in function, okay? So I sort of think about proteins as sort of like a lock and key, right? They're all designed to go do something, and that structure is important. So if that's our protein and that's our target, we need those two things to fit together. Their structures need to be compatible. So we might also have proteins that bind to the DNA or do something similar. So if we go in and introduce a mutation, in this case I'm going to delete the last exon. We delete the last amino acids on that protein, and we remove part of that protein structure. It's no longer there. So now at this point, if we try and have that protein do what it's supposed to do, it no longer sort of fits with whatever it's supposed to do because we've changed the structure. Okay, so when all goes well, we have the sire and the dam, and each of them are going to have a set of chromosomes. All right, so they'll each have two, one from their mom and their dad. And one of those chromosomes will randomly end up in either a sperm or an egg. That sperm and egg come together, and we produce a calf, okay? Another scenario is that if we have a carrier, in this case, the bull's the carrier of whatever this particular mutation is. If that allele, or that chromosome carrying that allele, happens to make it into his sperm, we don't have the same issue with the dam, right? The chromosome that assorted into her egg is going to be completely normal. Okay, so those two come together. We've still got one functional copy of that gene, okay? So in this case, live calf, all is well. But if we have two carriers, and the chromosomes that harbor that same mutation that is allethal both end up in the gametes that end up in that particular calf, we have a problem, right? And in this case, I've given you an AM calf, all right? But the scenario if we're thinking about an early embryonic lethal is going to be very similar. So if we have either a homozygous normal or a carrier, we have a calf, and we end up with a calf on the ground at a later point in time. But if they happen to be homozygous for an embryonic lethal, the cow's open, and we don't get a calf on the ground, right? So one of the ways that we can try to identify these types of lethal alleles is to try and find mutations in the genome that we never see as homozygous in a live animal, okay? So that's basically where this project is going. So the first step in this whole process is to sequence a bunch of animals and try and identify any changes in the genome that are predicted to have a functional change. So maybe changes the structure of a protein or something similar to that, okay? And also, we can use information from other species like humans, mice, zebrafish, any kind of model species where we've seen that this gene is lethal if it's knocked out in those species. That gives us some additional information that it might be lethal in cattle as well, okay? So once we have those variants identified, you can make a genotyping chip, put the candidates on it. From there, you really need to genotype a very large number of animals. So in this case, the goal was to genotype 10,000 heifers. And they don't necessarily have to be heifers, but the advantage of using those heifers is that we have additional information from their reproductive data, which can also be used for other types of purposes to sort of get it fertility in an alternate pathway, okay? So from there, you take a look in that larger population where we have sort of the candidates genotyped. We can see, do they ever turn up as homozygous in that population? And if the answer is no, they're a candidate to be a lethal, right? But the other thing we need to think about is allele frequency, and this was something that was talked about earlier today too. So if we think about something that's very, very rare at 1% in the population, we would really only expect to see one homozygous recessive individual in those 10,000 animals, okay? So if we don't see any, that doesn't necessarily mean that that's for sure a lethal. It means that we need to genotype more animals so that we can be sure for any of these things that are really, really low frequency. So as you might imagine, this is sort of an ongoing process. You identify these candidates, and you have to sort of continue collecting information on them over time so that we can verify that, yeah, we don't actually see those in homozygous form. So hopefully the way to do that is to place promising candidates on commercialized genotyping chips, okay? So you can genotype a large number of animals per year and have greater confidence that you've actually truly identified a lethal. Okay, So there are some limitations to this approach. The first of those is that you're limited by the number of animals you've sequenced, okay? So in that initial sequencing step, when we're looking for variants that may cause a protein conformation change or be lethal, right? If it's not a variant in that original population, we don't have that as a candidate when we go to those later genotyping steps, right? So the key is to get as much sequencing data as you can, and it's likely as more and more animals are sequenced within the industry that more potential candidates would be identified, which could go through that same type of process. So the second limitation is that really we're looking at variations in genes for this particular project. So there's lots of things that are not actually in a gene that are really important to the function of a gene or the regulation of how genes are expressed, okay? So that's actually the vast majority of the genome, okay? So in this case, we miss anything that might have a regulatory function but isn't actually within a gene. It's also dependent on the quality of the genome sequence. Because we're shooting for things that are in a gene, if a gene isn't annotated, meaning we don't know where it's at in the genome, or it hasn't been identified, we're going to miss that with this type of approach. So improving the sequence assembly is going to improve our ability to identify any of these types of variants. Okay, so if we're going to validate, I talked about genotyping being a major step in that process. So all this is related to needing to really test these things in a much larger population. So the genesis of that is this GDP F250 chip. So that chip was designed partially with this project in mind to include a lot of these possible candidate variants. And I'll go through a few statistics on that chip here in a moment. But part of that was to genotype a really large number of heifers where we have this reproductive data to sort of narrow down that list of candidates. And also, we can use that information to be able to estimate the allele frequencies in a much larger population than just the sequencing population. Okay, so there were a lot of different data sources that went into the design of the chip. I'm not going to go into a whole lot of detail on this. But there was a large amount of genome sequence here on a wide variety of different breeds. So you can look and see if your breed of interest is on the list. But another part of this process is sort of error checking those variants. Because if you take a look at any variation that you identify in sequence data, some of that is going to be sequencing errors, and some of that will be actual variation. So having these other populations where you can see if that variant shows up in multiple different data sources helps you figure out what might be a sequencing error and what might be true variation that we'd be interested in testing for. So in addition to the original sequencing data set, there was whole genome sequence for a variety of different Indocene breeds. There was RNA sequencing data on 153 animals and 5 different tissues. Information from the Thousand Bulls Genome Project, and from dbSNP, and I believe a large portion of that was from the original genome sequencing project. Okay, so if you take a look at this table, I've tried to highlight some of the more important pieces of this table. The rest you're certainly welcome to ask questions on, but I just wanted to show the highlights. So one thing here is that AA. So that means amino acid substitution. So that's where I talked about earlier, where we have maybe a SNP, a single base change that ends up changing the amino acid sequence of a protein. It might change the structure of that protein. Okay, so that's what these are. And you can see there's a whole lot of them that ended up being included on this chip. So anything over here in this column was validated through 4 different data sources, 3, 2, 1. And then these over here were only ever seen in one data set, okay? But you can see a large portion of them actually did end up on the GDP F250, and part of that is that one of the goals of this project is to identify really low frequency variants, right? So there's going to be a number of sequencing errors in here, but there's also going to be a number of really low frequency variants that are actually important. There's also an imputation set within this chip, so you can impute between different types of genotyping products. Okay, a large portion of animals from this project were actually genotyped on that chip. looks like about 11,500. There were also animals genotyped from a number of the other projects that you heard about today, including feed efficiency and the BRD projects. Okay, so the only thing I'm going to say about this is that the sample call rate and the SNP call rates were generally pretty good, and that would be typical of this type of genotyping technology. Now, I think this slide is kind of interesting because what you see is if you look at the distribution of minor allele frequency on a lot of the other chips that we have available, it's pretty constant because there's an emphasis on sort of these variants that are at higher frequency. So for this, we were really interested in, for the purpose of this project, those variants that were at very low frequency, okay? So you see a much larger proportion of low frequency variants on the GDP F250. And you notice there's a number here that we're at an allele frequency of 0, and those would probably be those sequencing errors. So that relates back to this block up here that I highlighted that were only ever seen once, okay? And it turns out some of those are sequencing errors. Okay, so in terms of the type of mutations that were included in this chip, the amino acid substitutions were the ones I mentioned earlier. And here is an example of that, where we just have one amino acid subbed in for another because we had a single base change in that DNA sequence. So there are a few frame shifts also on there, and this slide illustrates a frame shift, and it's kind of cut off, so I apologize for that. But basically, if you have like an insertion or a deletion of a single base, you can shift that 3-letter code so that you get a different amino acid sequence. So again, something that's likely to change the shape and function of that protein. So here's sort of a structure, a structure of a gene, okay? So we've got 5' UTRs, we've got exons, which are those protein coding bits, we've got the intron pieces, they get spliced out, and the 3' untranslated region, and there are SNPs from both the 3' and 5' untranslated region, and there are also going to be variants within those splice sites, so where we sort of paste those protein coding pieces together. And then there are a decent number also that aren't within a gene. Okay, so the really interesting part of this is how many of them did we not see as homozygotes, okay? So you can see there's the setup on this is the number of variants that were genotyped, okay? And down here is the expected number of homozygotes based on the allele frequency. Okay, and what you see is when the expected number of homozygotes is very low, you actually a lot of times don't see a lot of homozygotes, which makes sense, right? These are things where we need to genotype a larger number of animals to really be able to determine whether they were a candidate lethal or aren't a candidate lethal. But some of these others are interesting, so I'm going to get rid of that bar there and zoom in on the rest. And what you see is sort of a strange bimodal sort of distribution, but it looks like these where we'd expect to see, you know, between 3 and 6 homozygotes, and we actually don't see any homozygotes, seem to be really good candidates for being lethals, but you actually see a lot of these over here where you expect to see a large number of homozygotes and you don't actually see any. So the real question is, what's up with these over here? Because there's such a large number of them, there's likely something else going on rather than the fact that they're just lethals. So one possibility is that they're SNPs that just don't genotype very well. The second possibility is that because of the sequence assembly, we may have things that are actually duplications. So you see a much larger number of heterozygotes and not very many homozygotes. Okay, so one possible reason for that, I've got 2 sample chromosome 15s here. Okay, and if we have one gene that's been duplicated a couple of different times, and then those duplications can slowly change over time. So we might have a C here, and an A. So we had a mutation in that last duplicated gene. And a C, an A on the other chromosome. So he's actually homozygous at 3 different locations. But if that's a duplication that would design the same on the assay, you might actually get everything to genotype as a CA heterozygote. and never actually see any homozygotes, even though they are homozygous. Okay? So there's some that may be a result of that. There's also at least one here that seemed to be a fairly common haplotype that was never actually seen as homozygous. Okay, so as you can tell, we're a couple years behind where the other projects are because it was awarded a couple of years later. So there's a lot of work still ongoing in this project. So the next steps are really to narrow down and try and identify the candidate lethal mutations using a couple of different methods. Hopefully, finding some good candidates, those could maybe be incorporated into commercially available genotyping tests because this does need to be a continuously sort of ever-evolving process to get the most benefit out of it. And then the next question is, okay, if we identify some of these lethals, what do we actually do with them? How do we manage these sorts of things so that we can avoid causing any early embryonic mortality? So I want to give this a little bit of perspective. This is a slide where the blue dots are possible lethal mutations, numbers of possible lethal mutations, okay, in a couple of different animals, all right? The red dots are the potential impact on fertility, okay? So the one I want to point out are these two bulls over here that actually have a fairly large number of these potential candidate lethals in this fictitious data that we're looking at here, okay? So you can see that one of them has a very large impact on fertility versus the other one, his lethal alleles have a much lower impact on fertility, okay? And a lot of that relates back to allele frequency, right? Because if we have a bull with a mutation that's lethal, right, but and he's carrying that, and nobody else in the population has that lethal, does it matter? No, it doesn't matter, right? Because we can't ever mate him to another carrier. So we never produce any affected offspring, or we never lose any pregnancies as a result of that. So the frequency in the population is going to determine the impact on fertility. Things that are very common in the population are going to be things that have to be managed around to avoid these types of issues. Okay? So before I turn it over to Allison, I want to give a little bit of a summary. So the GDP F250 was designed as a research tool to meet the needs of a couple of different projects. And it is gene-centric, but it allows imputation into other types of data sets. Only about half of the amino acid substitutions that were detected were able to be designed on the assay, but it did contain every amino acid substitution that was designable. And it is publicly available now through GeneSeq, as someone else mentioned earlier today. In terms of the lethal variants, sort of the number of candidates right now is about 2,000, but lest anyone panic, that number's high, right? Not all of these can be lethals, okay? So this again goes back to that ongoing process of narrowing these things down and really filtering all of this data so that the maximum amount of information can be pulled out of that data set. So this is going to be evolving over time as this project proceeds. So two of the things that are coming in the future are really about delivery to the industry. But before I go and talk about that, very quickly I want to point out that there is a website for this project. It's beefreproduction.org, and it's going to be a little bit different than the other project websites you've seen. It's got some links back to the eBEEF.org fact sheets. It also has some videos on some of these topics like genetic correlations, what are lethal alleles, those sorts of topics. So if you're interested in learning a little bit more, I'd encourage you to go to that website and check out the videos. Those are still coming, so there's not a lot posted at the moment, but there will be more and more coming in the future. So in terms of use of this information in the future, one of those possibilities is to develop selection indexes that support multi-trait selection that includes these fertility-related traits. And Mike McNeil is going to be working on that. And I'm not going to belabor the last point very much because Allison can tell you more about it. But one other option is to develop decision support software that really allows optimization of not only your matings in terms of genetic merit, but also managing some of these matings for carrier to carrier matings. So with that, I'm going to let Allison tell you all about it and demonstrate it.
Thanks, Megan. So I'm just going to have a couple of slides here and then go over kind of trying to demonstrate to you how mate cell works. And there it is. So basically the rationale behind mate cell, which was developed by Brian Kinghorn at the University of New England, is to try to get all of the things that have to be considered in making mate allocation decisions, that is, which specific bull will I mate with which specific cow, all into one program that makes the best decision based on the information provided, but enables the breeder to go in and tweak the result based on the art of breeding, I guess. And so you get a result using mate cell, and then you can go back and craft exactly what it is that you want. And so it basically integrates complex breeding issues into a single. Well, Brian says it's easy to use, but Brian is not your average Joe. It takes a little getting used to, but it's actually a very powerful tool for making optimum mate selection decisions. And basically, as I'll demonstrate, you feed into mate cell your herd file. And by your herd file, I mean all of the ancestors of your animals. This would be basically what's maintained at your breed association. All of your active females that are on the ground with legs that need to be bred. And then all of your potential sires, be they AI sires or sires you have on site. And in there goes all of their information related to their EPD values for all of the different traits. And then you say, okay, I've got 100 cows and I've got 10 bulls, and I'm working towards this particular selection index. I'm going to choose $beef because that's the one I've just demonstrated here. Or maybe you'd use an index that includes fertility, like we're planning to do with this project. And mate cell will go through and make every single mate allocation of each bull with each cow, and it will get to an optimum where you've maximized your rate of genetic gain whilst minimizing your rate of inbreeding. And depending how aggressive of a breeder you are, if you want to just forget inbreeding and just mate every single cow to the very best bull there is in the breed, it will let you do that. Or if you're very averse to inbreeding and you don't ever want to use the same bull, it'll let you breed a different bull to every cow. So there's a lot of flexibility in there in terms of how aggressive of a strategy you want to use. And so that's kind of the way that it works. And the reason that I thought this would be an appropriate tool for this project is it allows you to do genetic defect management. And so typically what's happened when there's a carrier animal is the strategy is like, oh, kill it, get it out of here, never going to use that again. And as we've seen, and I think 2,000 is an overestimate, but I think we'll find every animal carries a couple of deleterious alleles or undesirable alleles. And so we can't just get rid of them all because we won't have anything left. And so you can actually control the likelihood of getting an affected animal, the little a animal, by just making sure you never actually mate two carriers, right? Because then you'll never have the opportunity for little a to come together. And you can control these recessive alleles using intelligent mate allocation rather than getting rid of the animals. That's kind of the rationale. And this slide here, I don't know, Matt, these lights here, can they go off, or is that just these ones? Okay, all right, never mind. Basically the only time, in this particular case, we're talking about a recessive lethal, so if you get little a, you're dead. So this never happens, this never happens, this never happens. The only time you get an affected animal, this black animal here, is when you have two carrier matings. So if you can manage a carrier by mating him to an individual who doesn't carry it, then you never have an affected offspring. And if you know the carrier status, you can manage them appropriately. So that's kind of the rationale behind the program. And what it does is it gives you the right balance between your formal breeding objective, and that's for you to just define. And in this case, I used $beef because it was in my herd, in my data file that I got from the Angus Association. And then I can do additional constraints. I can say no carriers can be used, or I don't want any affected individuals. In other words, I never want carriers that are at the same loci to get mated together, but I don't mind using carriers in my breeding program. I just don't want affected calves. And it kind of also, what I think is most compelling, shows you the opportunity cost of imposing non-optimal constraints on mate selection. So if I say, I'm never using a carrier ever, 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 you can see what that does to your genetic progress. And it is actually quite interesting to look at. And so, oops, let me just see, did I go past? No? No, that's not what I wanted. I've lost the slide I wanted. Basically, oh no, this is kind of it. So we simulated a herd. It was actually not a simulated herd. It was the Carsten's herd. He let us have his herd file. And then we pretended there were 6 minor, not very high gene frequency recessive alleles floating around in his population. And we used a simulation program to give different animals different alleles at different frequencies. And then we had 169 female selection candidates available and 85 male selection candidates that were available. And we had to make 125 different matings. So we had to do that. And then I'm going to show you what that looked like, and then I'm going to come back to this because this is when we started dealing with the recessive lethals. So I'm hoping this program's going to work. Looks good so far. So this is not running at the moment. I just want to give you a feel for it because it's a little bit like when you first see it, but calm, relax. So what this shows here is what's called the frontier. And basically, unfortunately, I've cut off the edge of mine also, Megan, but on this side is genetic, is dollar beef genetic merit, basically. So this is 0 and this is like 100. So getting better here. And over this way is inbreeding coefficient. So small amounts of inbreeding here, large amounts of inbreeding over here. So basically what happens is I say, okay, go ahead and make my 125 matings, and it does the calculations looking at all of the available cows and all of the available bulls. And it comes to this solution here where it's picked the right bulls and the right cows to give me the average genetic merit index of the offspring being here. And what you see here is all of the different components that are in the data file that I've fed in there. And so there would be some familiar data here. So I can see calving ease direct. Here's birth weight, here's weaning weight, yearling height, scrotal circumference, and so on down here to some of the other indexes that are in there. And all of this is being driven by dollar beef. So let me go ahead and run the program so I can kind of give you a feel for how it goes. So I'm going to push run here, and what it does sometimes when it doesn't crash is, oh, that's not good. Okay, all right, well, it's not going to do it for us today, sorry. Basically, what happens here is this is what I was saying is the breeding objective over here, and this is the inbreeding rate. And what happens if I say I'm not at all worried about inbreeding, my only interest is maximizing my rate of genetic gain for dollar beef, then I'm going to be over here selecting only the very best AI bulls for all breedings. And over here, I'm very disinterested in my rate of genetic gain. I'm very worried about inbreeding. Here, I'm going to use many different bulls from many different families. And this frontier here is kind of the equilibrium point, if you will. So it's kind of balancing inbreeding and rate of genetic gain. And you typically want to be somewhere in here where it gives you a solution where you're making good genetic progress, but you're not using just one single bull. And basically at the end of the day, what comes out is you'll typically get maybe 4 or 5 of the best genetic bulls being used and getting mated to the least related females. That's kind of what the typical result comes out as, is you get 5 or 6 of the best bulls getting used with certain specific females. So what we did on top of that then, going back to this slide, is we also added in here these 2 additional traits. So this trait here is lethal A, and that's the predicted number of recessive lethal alleles in the progeny. And so lethal alleles is not an issue. What is an issue is lethal genotypes. That's little a, affected individuals. So you can manage it to really minimize the number of alleles in the offspring, and that's kind of avoiding the use of any carrier animals in your breeding population. Or you can try to optimize it to minimize the number of recessive affected animals, little a, which allows you to still use carrier animals, but to use it in a way that avoids them from being mated with a heterozygote at the same loci so that you get an affected animal. And so whether you choose this strategy, that is never using a carrier, or this strategy, trying to never use a carrier that's also getting mated to a carrier at the same loci, really dramatically affects your rate of genetic gain, because this, if you have a reasonable frequency of the recessive lethal alleles, many of your animals are going to be carriers, and it's going to have a really hard time trying to minimize the alleles in the offspring, and you're going to dramatically tank your genetic progress. This is a much more conservative strategy that avoids affected offspring, but it still enables you to make genetic progress. And so hopefully my computer will agree with me that it's going to go ahead and show you what that looks, what that looks like, because I do have a movie embedded in here that I think will work. And basically what you're going to see is you're going to see this frontier getting formed, and then you're going to see me go in on the screen and say, okay, now I want to decrease the number of either alleles in the offspring, which is again basically not using any carriers, or minimize the number of affected genotypes in the offspring, which is basically trying to avoid affected individuals. And what you'll see is it'll be up here at the frontier, and if I make a decision that's too non-optimal, that is, I never want to use a carrier, you'll see what happens is the rate of genetic gain will tank. You'll see this little red line go down to here, and the matings it chooses has complied with your desire, whatever your desire might have been, maybe not using carriers, but you've gone from having an average index of $beef at 75 or whatever, down here to 60 or something like that. And so you've got this very big decrease in your genetic gain, which you can kind of balance against, okay, I didn't have any dead calves or curly calves or whatever, but every single mating I've made has been charged $25 for that, and actually I only would have expected to get maybe one, and so I've made kind of a suboptimal decision. That's kind of how this works. So let's see if this program is going to show us what this looks like. So now I have to find the movie. So here I've started the program, and I'll just stop it real quick. So you can see here we've got all of the different traits moving here, and so calving ease direct and birth weight and everything. And you'll see it going up here. So now it's kind of hovering at that optimum solution. That's the balance between inbreeding and genetic gain. Okay, so it's kind of set there, and I'll stop it for a second. If I went and said stop and went and looked, it would give me the breeding list that I would give which bull to mate with which cow to get that rate of genetic gain, and on average my offspring merit would be 70, whatever that is, 75. What I've done now is I've blown up the little square that's lethal G, that is the affected offspring, the calves that are going to die because it's lethal. And you can see it's not zero here. So there's a small number of calves with a certain probability that are going to get little a. So 0.25 here is going to, so on average 0.25 calves will get little a, and we'll get this one here that's affected, this one here, so these are less than 1. And then the majority, if I'm not doing anything to control for these recessive lethals, won't actually inherit little a because it's not these are not high frequency. There's 6 of these lethal alleles. So I'm going to let it keep playing, I think. Okay. Where are you? Okay, so what's happening there is I'm now saying, okay, I want to decrease the mean, and I'm giving it a very heavy weighting. So I want to decrease, I want to get rid of all those lethals, and so I'm no longer getting affected animals, but my average rate of genetic gain has dropped down here, and this is the optimal, this is the best it can do when I give it that constraint. So I've really dropped it. If I now go in and make that not quite such a heavy weighting, then I get to a solution up here where I've got very few that are getting impacted, but I've dramatically increased my average genetic gain from my mating. So this is a better solution probably for that little number of recessives. Now I'm going to go in and say I want to try to, I've released that hold. So that's what happens if you don't control for it at all. And now I'm going to go in and say, okay, now I'm not actually going to use any animals that are carriers because I want to reduce the number of alleles in my offspring from these matings. And that's what I get in the absence of doing anything. Now I'm going to say I want to decrease the mean for the number of alleles in the offspring of these matings. So I'm going to try and decrease that mean. And when I invoke that, you'll see this precipitous drop, and it comes to an equilibrium here. It can never get zero because every animal has, one or two of these, and so. But I've made this tremendous, dramatic decrease in my genetic gain, which when I release that constraint, pops back up to the solution here, and you get these, a certain number of alleles there. If I now just go in and put a little bit of emphasis against actual lethal animals, genotypes again, then I get this much better solution with a very small probability of getting one or two calves that die. And so that kind of mechanizes or gives you an approach to kind of play around with things and say, okay, this is what's important to me. And I could have equally gone in and said, okay, actually what I'm interested in is I could do something crazy like I'm only interested in really dramatically lowering my birth weight. And you can do that, you can tell it that, and it'll show you how that affects your overall rate of genetic gain towards your breeding objective. And so it really lets you play around with what are the implications of having maybe truncation selection for a certain marbling level, or really trying to decrease or increase one or two different traits. And you can go in and play with that and get different solutions. And so when we get the data from Jerry, and I'm hoping it's more like 6 rather than 2,000, but we have modeled this with 100 different traits, and there's really no way you can intelligently manage that number of recessive alleles without a computer program like that. There's just too many for the human brain to do all of the different matings that might occur there. And so what we hope here at the end of the day is to get out a program that takes in your herd file with all of the different EPDs of interest, that has an index of interest, and allows you to make optimum mate allocations to maximize your rate of genetic gain whilst minimizing the occurrence of affected offspring using computerized mate programs. So I know it's a little complicated, but as we get the data, hopefully it'll become a little bit more obvious what we're trying to do here. And so the bottom line is, I think every animal's going to be a carrier. We know we are all carriers, so I think that that's likely to be found out in cattle too. And that selection against matings that result in little a homozygote progeny resulted in a much better outcome than selecting against carriers or selecting against getting A alleles in the offspring. And ideally, the economic weighting that's associated with embryonic loss should be incorporated into the index to ensure that the optimum compromise is in there. At the moment, if you put too much weighting on that, then you drop your genetic merit progress much more than the actual cost of that particular recessive allele, and that's not an economically optimum solution. And so ideally, the objective is to identify lethal recessive alleles and develop an implementation for strategic mating and ensure that carriers are mated in a way that minimizes affected offspring while still utilizing the excellent genetics that comes in every other loci that animal's carrying. And then ensuring that the discount that a carrier gets is proportional to the impact of it on the economic progress of the herd. So with that, I think I'm maybe 5 minutes over, but oh well. I'm happy to take questions, and I'll finish there.
Questions for Allison? Or Megan, sorry. Questions for Allison and Megan? You can ask them all Allison. I guess I'd have one relative to the mate selection software you showed.
Yep.
How is, or what is the plan for that being released to the U.S. beef industry?
So that's really something that the breed associations would have to discuss with Brian Kinghorn, who's an emeriti professor at University of New England. I know that we're going to go and talk to Angus in a couple of weeks here to kind of talk about, because this project actually was kind of designed with that the 1,000 or 10,000 heifers were all Angus heifers, and so that's kind of where we were going to start. But obviously the breed associations are going to have to want to use the software and have it available on their website or something like that, and make it a lot less clunky than the one that's on my my laptop, so that it can actually be used. But this is getting used by breeders in other industries. It's used quite extensively in the pork industry and quite extensively in the chicken industry to make optimum selection decisions for those industries. And so I think there's a definite indication that it's got a lot of value there for those industries to adopt it. When I see them adopting it, that makes me think that it's good. It makes sense, and so I think that's the plan. But it's really up to the breed associations to determine whether they think this would be something useful for their breeders.
Other questions for either one of these two speakers? Arlen?
Do you foresee that this data be presented on a herd basis opposed to an individual basis?
So the way the program works is you can do it on an individual basis, which I'm imagining how seed stock breeders would like to use it, or you can also have a herd bull strategy. So I've got 100 animals and I need to pick 4 herd bull, or four herd bulls, and it'll go through and optimize that so that it's on average the best selection of 4 bulls for that particular 100 females, but it obviously won't say this bull has to mate that cow because it's a group mating situation. So there is, because this is also used in the sheep industry where that's the norm. So it can be made more commercially relevant as well. And you can even have it so you don't actually know the genotypes of your animals, but let's just say you've got a herd of one particular breed and we know the allele frequency of that particular recessive allele is 0.05. And I bought a bull that's a carrier, then it'll work out statistically what the likelihood is of getting affected animals based on that breed average for that particular allele, and you can compare that bull to maybe a bull that's not a carrier that has a lower genetic merit, and it'll do the math and figure out which is the economically optimal bull to, is it better to have a high merit bull and 2 lost pregnancies, and the next time round it might catch because it didn't happen to get double recessive. So it kind of does the math on that, and you have to have a kind of an economic weighting of what does it cost to have a pregnancy delayed from the first breeding to the second breeding, and then you've got another 25% chance of it happening the next time, and then again the third time, and then maybe she goes from the herd. So it's quite an economic thinking as to what exactly the costs are of having a carrier animal in there, but it is designed to also be used in a commercial setting if people are interested in doing that. But to me, its main utility and the real value is in the seed stock sector where you're doing individual matings and you can just basically get the best mating based on this data.
Yeah, that's a great question. I don't think just from, and Allison, you can weigh in here if you'd like. I don't think just from the identification process we would necessarily know when that would likely occur. There might be some clues in the data from, if it's something that's been shown to be a lethal knockout in mice, or something like that, there may be some clues to sort of when that might take place. But I think that would require some additional digging into a specific mutation to sort of figure that out.
Because to go into what Allison's saying, you would have to know that for Brian to be able to program that in.
Well, I think if it was anything that happened sort of before birth or even at birth, I guess you could include that, but you're right that there may be some varying economic consequence in when that occurs. And I don't know, has Brian accounted for that, or is there a way to account for that in there?
I think your question, Dara, does it happen day 0 or day 2 or day 30 or day 60? Because 60 has a different impact than day 2. And absolutely. And I think the only data we have really to look to that is the phenotypic data we have on the heifer, the 10,000 heifers, where we actually do have when the AI straw went in and whether she was pregnant very early on and whether she delivered that calf, we'd be able to see, there's some double recessive that or presumptive double recessive where those cows were pregnant and then showed up open at 90 days or whatever. But boy, if we're, I think more generally it'll just be kind of an estimate of kind of the average cost. I don't know that we'll get down into the weeds quite to that level, especially if we're talking about 20 or 30 loci. I think we'll just take on average that they die in the first 2 days, and that it's a really early lethal recessive. So do you want to?
Other research, do you have any?
Yeah, I mean that's going to be the same as the kind of environmental background in the bovine respiratory disease project, where you have an animal that lost a calf because, I don't know, she got moved at 3 days instead of letting her rest, and it'll appear as if she aborted, and we'll say, oh well, that was due to genetics, when in fact it was environment. So that'll just kind of be genetic noise in our system, and there's really, with 10,000 commercial heifers, there's no real way to control for that particular thing. So there'll be noise in the background, no doubt, but hopefully we've got enough animals that we will actually see when there's real recessive lethals that are missing. When you get really low frequency and you're expecting one and you don't see any, if it's that low frequency, I'm not too worried about it. I think the ones that worry me are the ones that are at a frequency of 0.1 in the herd or something like that, because then that actually could have some pretty impactful economics, and fortunately they're the ones we're most likely to see, because we're expecting in 10,000, if it's 0.1, you can kind of do the math, and it's going to be a number of animals that we should see that have had homozygous little a that aren't present in the population, and hopefully the environmental noise won't be enough to swamp that out.
All right, well, we've got, okay.
I just want to say one more thing relative to Allison's comment on your question, Dare. So that's one place where breed association data also might play a big role in that in the future. For breeds collecting data for like heifer pregnancy and stuff like that, the odds in those 10,000 heifers of getting two of the same carriers together might be pretty low, but over a large number of animals over a long period of time, that might be another way to get at that information.